In this video, I'm going to be summarizing the two topics, mutations and gene pools, together. Now, I actually just spent about 35 minutes recording this video, but I hadn't actually pressed the screen record button properly. So I'm sorry if I rush through this video a little bit. All right, now these here are our syllabus dot points for the two topics. And then these are the subtopics we learnt about, okay? So with mutations, we learnt about what mutations are, we learnt about what variation is, and then we learnt about types of mutations. So somatic versus germline and gene versus chromosomal. Finally, causes of mutations and examples. With gene pools, again, we learnt about what gene pools are. We learnt about what random genetic drift is and how it can cause changes in gene pools, and then what gene flow is and what barriers to gene flow are. Now, starting with variation. So variation basically just means that we all look different, okay? So variation exists amongst individuals of the same species, where we are different in terms of our genotype and phenotype, okay? So we're human beings, we all follow the same genetic code, we all have the same genome. But the reason that we don't look identical and act identical is because of this variation here. So there are changes in our genomes, meaning that our phenotypes or our physical expressions of the genomes are different, all right, and so therefore we're different. Now, one source of this variation is mutations. A mutation being a change in the genes of an organism, okay, resulting in permanent changes in the genotype and the phenotype of that organism. Now, a mutant is an organism with a characteristic resulting from mutations. This is the first way in which we can classify mutations. A somatic mutation, remember, occurs in body cells, okay? The gametes are not affected. Now, because of this, only the individual with the mutation is affected, all right? So let's say you have some UV damage to your skin cells. Those skin cells are going to be mutated. That mutation is going to be passed on through mitosis, okay? And so when those skin cells undergo mitosis and make more skin cells, they're going to make more skin cells with those mutations. That's actually how skin cancer is caused, all right? So the body cells are affected, only the individual is affected therefore, and it's passed on through mitosis. Now, on the other hand, we've got germline mutations, okay? So these are mutations in the gametes or the sex cells or the sperm or the eggs. Now, the individual is not affected here, but the offspring that the individual has are. So let's say that there's some sort of mutation occurring in a sperm cell of a man. That man reproduces, that particular sperm cell is successful in fertilizing an egg, okay? That offspring is going to have that mutation that was carried in the sperm cell. So therefore, these types of mutations are passed on through meiosis, and that's because that's the process whereby gametes are duplicated, all right? So a somatic mutation occurs in a body cell, only affects the individual and is passed on through mitosis, whereas germline mutations occur in the gametes or the sex cells, okay, therefore affecting the offspring and passed on through meiosis. Okay, now another way in which we can categorize mutations is according to the level at which they're occurring. So we can talk about mutations as being either gene mutations or chromosomal mutations. Now, with a gene mutation, we have a change in nitrogenous bases resulting in a single gene only becoming mutated. Okay, so we're having a very small change in that nitrogenous base sequence. A very small change, however, can have a very adverse effect. Two types of gene mutations. Firstly, we've got frame shift mutations, and then we've got point mutations as well. Looking first at our frame shift mutations. This is when the number of bases is changed, okay? And so therefore all codons onwards are different. meaning that amino acids added to the protein during translation is different. So I'm going to use this diagram down here to explain that a little bit more. Our first type of frameshift mutation is an insertion. So as you can see here, in our original DNA strand, the sequence is CTGAGT. Now, our second DNA strand reads CTGATGT, okay, and so we've had this T inserted here. Now, codons, remember, are triplets of bases. So this here is one codon, this here is one codon. This codon is CTG, this codon is AGT. Now, looking at our second DNA strand here, our codons have changed. We now have CTG and ATG. Okay, so this is different to this. This means that when the amino acids are strung together during protein synthesis to form a chain, the sequence is going to be different, okay? Or the sequence of the amino acids is going to be different. So that's how an insertion works. Now with a deletion, a base is removed instead, okay? So looking at number 1 and number 2 here, this A nucleotide is being deleted. And so instead of our codons reading CTG, AGT, we're looking here now, okay? We've got CTG, so the same codon as above, but our second codon only reads GT. And so that means that when those amino acids are put together in protein synthesis, we're going to have a different protein being produced. All right, looking at point mutations now. So this is when one base is exchanged for another base. We're not deleting or inserting a base, but we're exchanging them. A point mutation is also known as a substitution mutation, and that's because we're exchanging or substituting bases. Now this results in one different codon, which is translated into a protein. So again, because we're changing the codon sequences, the protein is going to be different. Now, there are 3 types of substitution mutations. We've got missense, nonsense, and silent. Looking first at missense. Now, you don't need to know the difference between conservative and non-conservative, so I'm going to put a cross through that column there. But you do need to know what a missense mutation is. Okay, so this is when the new base changes the protein from being produced. So in this column here, we've got the normal gene, okay, where there's no mutation occurring. We're going to compare this gene here, or this codon here, TTC, with this one here. What's happened is that, as you can see, that middle T has turned into a C instead. Changing this codon here, AAG, to AGG. Changing, therefore, this amino acid lysine to arginine, I think that is, okay? And so that means that instead of the lysine amino acid being added to the protein chain, an arginine is added instead, all right? So we're having a different protein being produced. Now, in a nonsense substitution mutation, we're actually stopping that protein from being produced. So we've had this mutation here, okay, where the T has turned into an A instead, meaning that at the mRNA level, our new sequence is UAG, meaning that at the protein level, we're actually coding for a stop amino acid, meaning that protein synthesis is stopped. Finally, with a silent point mutation, we've had this mutation here, okay, so the C has been substituted by a T. Therefore, at the mRNA level, we've got an AAA instead of an AA and a G, okay. We still, however, have the same amino acid, lysine, added to the chain. And that's because both AAG and AAA happen to code for that same amino acid, alrighty. So A gene mutation means that there's a change in the nitrogenous base sequence. We have got frame shift mutations, which is when we're changing the number of codons, and so therefore, we're changing the number of bases, and so therefore every codon onwards. And then a point mutation is when we're substituting bases. All right, looking now at some examples of gene mutations. I'm going to read through this table quite quickly, okay, because this is something you can have a look at later on in your own time. Firstly, we've got Duchenne muscular dystrophy. So this is a single gene mutation, meaning only one gene is affected. Now, DMD occurs through a mutation in the mother and is inherited by her sons only. What happens in someone that has muscular dystrophy is that the leg muscles waste, okay, or basically they kind of break down and they don't have normal motor function. And then later on in life, the arms, the shoulders and the chest muscles start to waste away as well. Now, eventually the respiratory muscles fail, okay, and the sufferer unfortunately dies. Now, DMD is usually diagnosed at around 3 to 5 years of age, and individuals rarely live past the age of 25, and that's because by the time they get to that age, the respiratory muscles have failed. Cystic fibrosis, again, another single gene mutation, okay, specifically occurring on chromosome 7. Now, cystic fibrosis, remember, is a disorder involving a channel or a protein pump on the cell membrane. This channel pump, or this protein pump, usually pumps ions across the membrane. And so this channel is faulty, meaning that too much water is pushed across the membrane. This causes a thick, sticky mucus to be built up in the digestive system, the respiratory system, the reproductive system even, okay? And so the affected person suffers from persistent coughing, wheezing, pneumonia, and digestive and other problems as well, okay, due to this thick build-up of mucus. Albinism is next, okay, caused by a nonsense point mutation on chromosome 5. Now, going back to here, remember a nonsense point mutation is when the new base stops a protein from being produced, okay? Now, specifically here, we're stopping this protein from being produced, melanin. Now, melanin typically pigments the skin, the hair, and the eyes. So if sufferers don't have this pigment, they are going to be very fair, okay, and they're going to have red eyes as well. Albinism is disadvantageous because it causes vision problems, okay, and that person does not have the normal UV protection that people have when they don't have albinism, and so they're very susceptible to getting skin cancer. Sickle cell anemia is next. So this is a codominant point mutation. I will talk more about codominance in the evolution video. But essentially what happens here is that you can inherit 2 dominant alleles, 2 recessive alleles, and then a recessive and a dominant allele. If you have the 2 recessive alleles, you have got sickle cell anemia, okay, and so you can get quite sick. Now, when you've got this codominant point mutation, the gene responsible for normal hemoglobin production is faulty, and so a mutant form of hemoglobin is produced instead. This means that your red blood cells aren't nice and round like this, okay? Instead, they're sickle shaped, so they look like those crescent moons. This means that you can't carry enough iron in your red blood cells, and so sufferers are anemic. Now, sickle cell anemia is predominantly found in sub-Saharan African populations. And that's because the sickle cell trait, which is this codominant form, provides what we call an advantageous mutation, okay? Meaning that these sickle cell or these sickle-shaped cells here actually can't be invaded by the malaria parasite, okay? And so if you have these, malaria can't invade your red blood cells to as great of an extent as they'd be able to otherwise. And so you can't get as sick with malaria, okay? I'll talk more about advantageous mutations and codominance, like I said, in the evolution video. All right. Moving on now to thalassemia. Now, there are two types of thalassemia, alpha and beta. Now, alpha thalassemia is due to a mutation in the HBA gene on chromosome 16, okay? This reduces the levels specifically of the alpha globin chain in the hemoglobin. Now, with beta thalassemia, the mutation instead is on chromosome 11. And so the level of beta globin in the hemoglobin is going to be reduced. So alpha and beta hemoglobin, both resulting in abnormal hemoglobin production, but they're just different in terms of the chromosome with which the mutation is on and specifically which form of globin is mutated. Now, thalassemia is mainly found in Mediterranean populations, okay, so Italy, Greek, and Cyprus. Again, we have codominance here. So you can either have two dominant alleles, two recessive alleles, or a dominant and a recessive allele. If you've got two dominant alleles, you are a homozygous patient, okay, you're quite sick, and you need to actually receive regular blood transfusions or you will die. If you are heterozygous or you've got the codominant form, you've got an advantageous mutation, again, against malaria, okay? And so because you've got some of those sickle red blood cells, malaria can't invade your red blood cells as much. So again, in terms of symptoms, sufferers have less hemoglobin, meaning they can't carry as much oxygen, okay? And they can experience symptoms ranging from mild anemia to organ damage. All right, finally looking at Tay-Sachs disease. Now, Tay-Sachs disease is what we call a lethal recessive condition. This means that if you inherit two recessive alleles, then you are unfortunately going to die from Tay-Sachs disease, okay? It's fatal. Now, the mutation involves the hexa gene, which causes the disruption of this enzyme here, and I'm not going to try and say it. But what this enzyme normally does is it breaks down toxic substances in the central nervous system. And so without the breakdown of these substances, nerve cells die. Now, being a lethal recessive disorder, most patients with Tay-Sachs don't live past the age of 5, okay? And so therefore they're not going to pass those genes onto the offspring. Now, Tay-Sachs is quite prevalent in the Eastern European Ashkenazi Jewish population. And that's because this population is what we call a founder population, meaning that it started from only a few individuals. Now, how this has happened is that those few individuals that founded that population would have happened to have had many of those alleles within their genomes, okay? And so that means that when they bred with each other, they kind of interbred those recessive alleles into the population. Now again, Tay-Sachs can provide a slight selection advantage, okay, but instead of providing an advantage against malaria, it provides an advantage against another disorder, tuberculosis. So we just talked about gene mutations, which is when we have a change in the nitrogenous base sequence. Our other type of mutation is chromosomal, okay? This is when we have got a structural change. So this is when part of or a whole chromosome is changed or mutated. Now, we result in a number of genes being mutated here instead of just one. There are 5 different types of chromosomal mutations. Let's look first at a deletion. So in a deletion mutation, part of the chromosome is deleted, okay? So looking here, this DEF segment is being deleted, resulting in this much shorter chromosomal segment there. With an inversion, a part of a chromosome breaks off and it reattaches, but the wrong way around. So here, this DEF segment has broken off and reattached, but it now reads FED because it's attached the wrong way around. And so that means that when that nitrogenous-based sequence is read in protein synthesis, a different protein is going to be produced. Next, we've got duplications, which is when a section of a chromosome is accidentally copied twice, okay? So we've got CD here. This CD segment is copied and inserted, and so now we've got CD, okay? Here, with a translocation mutation, a part of a chromosome is being broken off and rejoining, but to the wrong chromosome, okay? So here we've got JK. It breaks off. Instead of reattaching to that same chromosome, it's actually reattached to this chromosome up here, okay? So you can see this segment has reattached here instead of where it should have attached, which was here. Now, with non-disjunction, we're dealing with meiosis. Now, remember that during meiosis, we've got our homologous chromosomal pairs that separate, one being pulled to one cell, one being pulled to the other cell. During meiosis, 2 copies of one chromosome go into one gamete, while the other gets no chromosomes, okay? And so the resultant cells are going to have incorrect chromosome numbers. Now, aneuploidy just means that the chromosome number has changed. So a cell resulting from non-disjunction wouldn't have the normal 46 chromosomes, okay? Well, 23 chromosomes because we're dealing with gametes. It might have 22 or 24. Now, looking at this diagram here, we have a cell that's about to go through meiosis. Looking at a normal separation example, we've got one red chromosome going into this cell, one blue chromosome going into that cell, okay? Looking at non-disjunction, both the red and the blue chromosomes have gone into the same cell, whereas here, we've got no chromosomes, okay? And so when this chromosome undergoes the second round of meiosis, again, we don't have any chromosomes. When this cell here goes through the second round, we've got too many. Now, this cell here and this cell here is a normal cell with only one copy of the chromosome. But if any of these other cells, so these ones or these ones, if they happen to be successful in fertilization and then making an offspring, or if they turn into an offspring, we're going to have a different chromosomal number and therefore aneuploidy. Now, when we have an extra chromosome, we say that trisomy has occurred. So we've got 3 chromosomes. When we're missing a chromosome, we say that monosomy has occurred and we've only got 1. Looking at some examples of chromosomal mutations now. Our first is Down syndrome. Down syndrome is also known as trisomy 21. Trisomy meaning that we've got an extra copy of the chromosome, okay? 21 meaning it's occurring on chromosome 21. So what we have is instead of 2 copies of chromosome 21, we've got 3. Now, Down syndrome results in a spectrum of defects, including mental disability, an increased distance between the eyes, and heart defects and other physical malformations as well. Patau syndrome is another example of trisomy, okay, but instead we've got 3 copies of chromosome 13. Symptoms include a small head, smaller than normal head, an extra finger on each hand, a cleft palate and lip, and mental disability again. Another example of trisomy is trisomy 23. Now remember that 23rd chromosomal pair is actually the sex chromosome pair. So normally, if you're XX, you're a female. If you're XY, you're a male. Now, sufferers of Klinefelter syndrome are technically male, okay, because they've got an X and a Y, but they have an additional X here. Symptoms include small testes and infertility because there's not normal sperm production, enlarged breasts, sparse body hair, and occasional mental disability. Cry-du-chat syndrome is next, okay, also known as cry of the cat, okay, and that's because babies that have this syndrome have this characteristic kitten meow-like cry. And that's because they've got problems with their larynx and with their nervous system. Now here, the mutation in which we're talking about is a missing part of chromosome 5, okay? And so we don't have monosomy or trisomy or aneuploidy at all. We're just missing part of a chromosome, okay? And so we've got partial aneuploidy. Finally, Turner syndrome. This is another chromosome mutation that's dealing with the sex chromosomes. Instead of having two X chromosomes, sufferers of this condition have only one, okay, because they're missing one. And so these, the people who have this condition are female, okay, because they've got an X chromosome. They develop no secondary sexual characteristics, they're infertile, and they're usually quite short in stature. All right, looking finally at how these mutations are caused. Remember that there are two different types of mutations in terms of how they're caused. We've got induced mutations and then spontaneous mutations. Looking first at our induced mutations. These are caused by mutagens in the environment. A mutagen being a chemical agent, so for example, mustard gas or formaldehyde. Now, mutations resulting from mutagens may be gene or chromosomal, okay, so they can occur at the gene level or at the structural level, and they can be somatic or germline, so they can occur in the body cells or the gametes. Now, how this works, how a mutagen can cause an induced mutation is because of this reason here, or these reasons here, I should say. So firstly, mutagens can resemble proteins and be incorporated into the DNA. They can trigger DNA replication errors. They can cause DNA strands to break or lengthen. They can block DNA replication or damage the structure of the DNA, all right, and finally they can chemically react with and modify DNA. So in all of these processes here, what's really occurring is that either the nitrogenous base sequence or the chromosomal structure is being changed in some way. And so during protein synthesis then, different proteins are going to be produced, okay? Now looking at spontaneous mutations. Instead of having environmental influences here, we've got random errors during biological processes, okay? So for example, mitosis and meiosis, all right? Cell duplication or DNA replication. Looking first at DNA replication errors. So DNA replication, remember back to year 11, is the process where new strands of DNA are made. Now, DNA replication is important because when our body cells replicate, we need to make sure the new cells are going to have the correct number of chromosomes. We can't replicate a cell without replicating the DNA. Now, during DNA replication, the DNA strand is unzipped and complementary strands are formed. So basically, we're making an identical copy. Now, errors can occur during this process, okay? These errors include frame shift mutations, okay? So those insertions or deletions, and we can also have substitution mutations, all right? Now, I'm not going to go over those mutation types because I just did, but basically, we're changing the nitrogenous base sequence, therefore the codons, and therefore the amino acids that are going to be strung together to form a protein chain, okay? Now, DNA replication errors can be either gene or chromosomal, and then somatic or germline, okay? So they can occur in both types of cells, and then at either the gene or the chromosomal level. Now, with our cell division errors, or meiosis and mitosis, we can only have chromosomal or germline mutations occurring, okay? So cell division errors can't occur in body cells or at the gene level. Now, during metaphase, normally we've got homologous chromosomes lining up across the cell, okay? And we have crossing over occurring, which is where genetic material is swapped. Oh, oopsies, let me go back. So looking at this diagram here, we've got chromosome 1 and chromosome 2 lining up. Now, as we have crossing over, they've exchanged some genetic material down here, all right? And so that's going to cause mutations, either inversions or translocations, okay? Now, in addition to that, we've got non-disjunction occurring during anaphase, okay, resulting in aneuploidy. So I talked before about non-disjunction in terms of how it's a chromosomal mutation. Remember that this is a type of spontaneous mutation because it occurs during cell division. So over here, with our induced mutations, we've got exposure in the environment to mutagens, okay? And then over here with our spontaneous mutations, we've got random errors occurring during biological processes. In the second part of this video, I'm going to be talking about gene pools. A gene pool referring to the set of genes within a population. Okay, so if you take a room full of people, let's say Miss Smith's Year 12 Human Biology class, all of their genes together is a gene pool. Now, we talk about populations when we talk about gene pools. And that's because a gene pool is in relation to a population. And we can define a population as being a group of organisms of the same type living in the same place at the same time. All right, so a population doesn't include, for example, humans and cats and dogs. It would only refer to the humans living in that place. Now, allele frequency refers to the percentage of organisms possessing a particular gene within that gene pool, okay? Now, variations in allele frequency occur over time, and I'm going to talk over the next 5 or so minutes about how these variations or changes occur. Now, natural selection, remember, is when factors within a population's environment cause certain phenotypes to be advantageous for survival or reproduction. And so therefore, those phenotypes eventually are going to increase in population. So natural selection is one of the mechanisms in which allele frequencies can change. Artificial selection or selective breeding can result in changes in allele frequency as well, okay, but to a smaller extent. And that's because artificial selection is the intentional reproduction of individuals in a population that have desirable traits, all right? And so this isn't something that we, I suppose, do very often or every day, and so it has a smaller effect than natural selection does on allele frequency. Now looking at random genetic drift. So random genetic drift can be defined as changes in the occurrence of genotypes in small populations due to luck. Luck being the key term here, okay? We don't have natural or artificial selection. We don't have evolution occurring here. We've got these random events causing changes in genotypes. These changes are random and non-directional, okay? And they're seen between generations over time. Two types of random genetic drift. There's the founder effect and then the bottleneck effect. Now, the founder effect occurs when a colony is started by a few members of the original population, okay? Now, this small population size means that the colony will likely have reduced genetic variation, okay? And that a non-random sample of the gene will occur, or of the genes. Okay, non-random. So looking here at this original population, we've got all sorts of different colours. We've got orange, green, red and purple. Now randomly, these 4 individuals have gone off and founded a new population. They have bred with each other, okay? Now in the future generations to come, as you can see, we've only got orange and green individuals. And that's because it just so happened that in this founding population here, we only had those alleles. this is completely random, okay? Because those individuals that have moved on to start that new population had random genes. Now, the bottleneck effect is similar to the founder effect, okay? Except the difference being that this is an extreme example of genetic drift because there has been some sort of event, for example, a natural disaster, which has severely reduced the size of the population, okay? So the allele frequency after the disaster or after the event is going to be different by chance from before the event. So looking here, we've got our original population. Now, just looking at this bottle quickly, it appears as though we've got lots of yellow lollies. Now, a random sample has been poured into a different jar, okay? So we have our surviving population here, representing the genes of that surviving population. After some time, those individuals have reproduced with each other. And then looking at the new population in this bottle here, it appears as though we've actually got lots of red genes and not very many yellow at all, okay? So we've had some sort of event that has significantly reduced the size of the population, resulting in the allele frequency being very different. So that's random genetic drift. Finally, looking at gene flow and barriers to gene flow. So gene flow refers to the movement of genetic material from one population to another. So when people move between populations, then gene flow occurs. Now, there are two types of migration that can occur. Immigration, which is when people move into a population, okay, and so therefore genes are going to flow in. And then emigration, which is when people move out of a population, okay? And so genes are going to flow out of that population then. Now, sometimes gene pools become isolated from other gene pools, which prevents gene flow, okay? And so we talk about barriers to gene flow here. And there are a few different types of barriers. Firstly, geographical, okay? So these are physical barriers in between populations that prevent people from moving from population to population. So for example, oceans or mountain ranges or lakes or deserts or ice sheets. Socio-cultural barriers, okay, aren't physical barriers, but we're talking about religion, social status, economic status, and educational background, okay? So people are choosing who they want to reproduce with depending upon these socio-cultural, that's a hard word to say, factors here.